To understand the deepest roots of late-onset Alzheimer's disease, we have to navigate to one specific, highly congested neighborhood in the human genome. For decades, the undisputed leader of this genetic landscape has been the APOE gene. Its epsilon-4 variant is the strongest common genetic risk factor for the disease, casting such a massive statistical shadow that it obscures almost everything around it. But if you look closely at a map of chromosome 19, you will find another gene sitting right next door. This is TOM40. Its endpoint is separated from the transcription start of APOE by exactly 2,000 base pairs, a microscopic gap in the genome. This proximity creates a persistent paradox. To a population geneticist, TOM40 often looks like a meaningless statistical echo of its famous neighbor. But to a structural biologist, the protein this gene builds is the ultimate choke point for survival. Reconciling those two views, making sense of that 2,000 base pair gap, is how we uncover the true bioenergetic driver of Alzheimer's disease. When researchers sequence large populations, TOM40 variants almost always travel on the exact same chromosomes as APOE variants. Because of this strict genetic tethering, statistical models routinely dismiss TOM40 as a passenger, a variant along for the ride without contributing to the pathology. Inside a living neuron, however, TOM40 is a physical machine. It forms the literal gatekeeper of the mitochondrion, acting as the sole entry point for almost every protein the organelle needs to generate power. To read this biology correctly, we have to separate the statistical locus from the molecular machine. We have to isolate what the gene looks like on a spreadsheet from what the protein does inside the cell. As we track both paths, we will arrive at a recent biological discovery that physically fuses these two opposing viewpoints back into one. Resolving the duality of TOM40 determines which molecular targets lead to effective therapies, rather than just marking the presence of a statistical risk. Roughly 2 billion years ago, the proto-mitochondrion was engulfed by a host cell. Over time, it surrendered nearly all of its ancestral DNA to the host nucleus. Today, this human mitochondrial genome encodes only 13 proteins. The remaining 99% of the organelle's working parts are manufactured outside, in the cytosol. That evolutionary outsourcing creates a severe logistical bottleneck. Every time a subunit of the internal energy grid takes oxidative damage, its replacement must be imported across the mitochondrial membranes. All of that inbound traffic converges on a single molecular machine, the translocase of the outer membrane. At the center of this complex is TOM40. Resolved at near atomic revolution in 2019, it is a 19-stranded beta barrel embedded in the outer lipid membrane. This barrel forms an aqueous conduit. Newly synthesized unfolded proteins must thread their way through this highly specific physical pore to reach the inner matrix. The entire supply chain of the mitochondrion runs through this single channel. Its fixed capacity means the organelle's lifelong survival relies entirely on keeping this narrow node open. While Alzheimer's focus is often on amyloid beta plaques accumulating between cells, the protein mounts a separate physical assault directly at the mitochondrial membrane. Amyloid beta peptides actually hijack the translocase machinery. They pass directly through the TOM40 pore, transit into the interior matrix, and physically bind to the internal electron transport chain, directly choking off ATP production. At the exact same time, amyloid aggregates wedge themselves deeply inside the TOM40 channel from the outside. They lodge firmly within the barrel, physically blocking the unfolded pre-proteins from entering. The damage compounds instantly. The mitochondrion is poisoned at its energetic core, while the specific supply line required to import replacement parts is completely sealed off. Amyloid beta acts as a highly specialized saboteur, shutting down the neuron's bioenergetic repair system precisely when it is needed most. This blockage triggers a devastating secondary failure in mitochondrial quality control. The cell uses a specific kinase called PINK1 as a surveillance sensor to decide if a failing mitochondrion should be destroyed through mitophagy. In a healthy state, PINK1 is continuously imported through the open TOM40 pore into the inner membrane, where it is immediately cleaved and degraded. As long as the pore is clear, the destruction alarm stays off. When amyloid beta clogs that channel, PINK1 can no longer enter. Instead of being degraded, the sensor molecules back up and rapidly accumulate on the exterior surface of the outer membrane. The accumulated pink 1 trips the alarm, but the signal is miscalibrated. Because the sensor is decoupled from the actual internal voltage of the organelle, the cell mistakenly flags a healthy, albeit blocked, mitochondrion for destruction, or fails to clear a genuinely dying one. This creates a vicious, closed loop. Initial bioenergetic stress generates amyloid beta. The amyloid beta clogs the import pore. The clog breaks the disposal system, and the resulting buildup of damaged organelles accelerates total cellular collapse. That mechanical reality makes TOM40 vital. But returning to the genetic map, we find a specific length polymorphism, a repeating sequence of thymidine residues, hidden inside intron 6 of the TOM40 gene, cataloged as 523. This polymorphism operates under strict linkage disequilibrium. They are inherited together. In populations of European ancestry, a long sequence is exclusively found on the exact same chromosome as the APOE4 allele. Because they travel tightly together, their statistical signals blend into a single massive spike on a Manhattan plot, blurring into one indistinguishable data point. Looking at that blended data, mainstream genetics concludes that TOM40 carries no independent weight and is merely a proxy marking the presence of APOE. A statistical correlation does not equal biological irrelevance. A gene can be mathematically masked by its neighbor while encoding a biologically indispensable machine. To prove TOM40 has its own effect on disease timing, researchers had to design a specific statistical filter to silence the APOE signal completely. The method is stratification. By systematically stripping away all patients carrying the epsilon 4 allele, researchers can isolate a purified group of individuals exclusively carrying the neutral APOE epsilon 3 genotype. Within this group, individuals with the very long variant develop symptoms years earlier and show accelerated gray matter loss. The length of this genetic repeat acts less like an on-off switch for the disease and more like an inherited dial. It modulates the expression of the TOM40 channel, establishing the baseline bioenergetic capacity for the neuron from birth. When the overwhelming noise of its neighbor is removed, TOM40 serves as the biological clock determining how quickly the neuron will succumb to the disease cascade. For 15 years, geneticists and biologists have argued across the aisle, trying to determine exactly which neighbor truly owns the risk signal on chromosome 19. A 2024 discovery regarding a transcript known as T9A2 provided a structural link that merges these two perspectives. Researchers observed read-through transcription. An RNA polymerase ignores the 2000 base pair boundary, reading straight across TOM40 directly into APOE, outputting a fused mRNA strand. When translated, the result is a structural chimera, the APOE protein tethered directly to the TOM40 beta barrel on the mitochondrial surface. The genetic boundary researchers spent 15 years trying to separate statistically does not actually exist mechanically at the surface of the organelle. This fused machine behaves differently depending on the inherited genetics. A chimera incorporating the neutral epsilon 3 variant provides a substantial boost to mitochondrial bioenergetics, while the high-risk epsilon 4 version fails to offer the same protection against oxidative stress. TOM40 is not a passenger to APOE. At the mitochondrial membrane, the import gate and the lipoprotein operate intricately as a single combined machine. This reveals a critical therapeutic limitation. Because a failing mitochondrion cannot import the parts needed to rebuild its own broken gates, clearing amyloid plaques removes the obstacle, but cannot restart the supply line. Future treatments must focus on directly supporting the gate's capacity and preventing its structural collapse. To halt the bioenergetic collapse of Alzheimer's disease, science must stop separating the statistics of the genome from the physical structure of the cell and address the narrow gate where the entire cascade begins.